Now, how do we go about inhibiting the function of an aberrant protein is what we created the SMNH platform for, where the SMNH molecule or compound is derived from the structure of a DNA or RNA. In nature, DNA and RNA interact with each other with exquisite specificity. And our concept is that if we can sort of mimic the interaction between RNA, DNA, and protein via an SMNH molecule, then we can get the kind of selectivity and specificity that we want in trying to achieve the inhibition of this aberrant protein. So that's what we have, our objective is. And in terms of making a drug to target an aberrant protein, there are, and to design an SMNH molecule, there are several concepts involved. For example, the molecule that we design and identify, it has to be made orally bioavailable. It has to be a pill that you can take. And that is very important from the patient perspective. That if you discover a drug against a disease, we just want to have it being made in the form of a pill that a patient can take on a daily basis. To make that happen, the compound or the drug that enters the system has to go to the target organ where this aberrant protein is, and then it has to selectively and very specifically interact with that protein. And so we want to be able to build the certain chemical features into that SMNH molecule that can achieve the desired effect of being able to be taken as a pill, it being able to go to the target organ, and then interact with that protein in a very selective manner, the aberrant protein. And we believe the SMNH molecule presents the ideal sort of platform. SMNH platform presents the ideal scenario for designing molecules, because we are basically mimicking the interactions, the natural interaction between a nucleic acid and a protein. And for example, here is a SMNH-like molecule which has the components which are required for the molecule to interact with the protein. It has all the essential sites or the bonding interactions that are required for this molecule to interact with that aberrant protein and inhibit its function. For example, it has hydrogen bonding sites, it has ionic interaction sites, it has hydrophobic interaction sites, and all these features are sort of inherently present in a molecule of which we design so that it can have that selectivity and specificity that we are trying to achieve. And besides this molecule has also, we can endow this molecule with the property that it can go to the selected target tissue, where it can enter the cell and then it can interact with that protein, aberrant protein. One more characteristic that we are trying to, we have been able to endow this molecule with is the fact that it can be made orally bioavailable. In other words, it can be made as a pill. And the other thing is, this molecule has the property that it is not going to be metabolized, or it is not going to be converted into some other molecule in the body, because it is designed in such a manner it doesn't have what is called metabolic liability, which causes a lot of problems with other kinds of molecules, traditional drugs. So this SMNH molecule, because of this all these unique features of being made into a pill, it can be target tissue specific, it can interact selectively with the aberrant protein, it doesn't have metabolic liabilities, and then makes this molecule sort of an ideal sort of platform for SMNH chemistry. And initially our target has been, as I mentioned, the antiviral targets, where we have discovered that this molecule can be used to activate the innate immune mechanism, which provides a very, what you call, pan-genotypic activity against hepatitis C, and activity against various resistant mutants against HBV. And basically what this molecule does is these type of antivirals which do not have the problem of resistance that develops with other kinds of antivirals, which are called direct-acting antivirals. This molecule acts on the host cellular machinery, and it only acts in virus-infected cells. That gives it the specificity that it is required. And so our first molecule, the compound that came out of this platform, is SB9200. And SB9200 is undergoing clinical trials. It has gone through a lot of discovery in terms of how we develop this molecule through screening in cell-based assays, and it has undergone in vivo efficacy evaluations, and then we have gone through a lot of preclinical toxicological studies with this compound, all of which is required as a prerequisite before we advance these molecules into human clinical trials, which is currently SB9200 is being tested in Australia in HCV infected patients. Our plan is to advance this molecule further into hepatitis B. This compound is also active, and once we complete the phase 1, we have a plan to take this molecule into clinical trials against hepatitis B. Both these viruses, Hep C, hepatitis C, and Hep B, are very serious health issues in infections with these viruses. Is one of the major problems worldwide. About 600 million people are chronically infected with hepatitis B and hepatitis C. It causes liver cirrhosis and liver cancer, and that's how the patients die from the disease. So there is a significant, very significant unmet medical need, because many of the current antivirals do not cure the underlying disease. They simply inhibit a viral replication.